Hi, everyone. I'm Linda Tintawi, CEO of the Lustgarten Foundation, and I'm so excited to welcome you to our very first Lustgarten Sprint to Discovery. Today, you're going to hear directly from our postdoctoral researchers, the next generation of pancreatic cancer scientists who are part of our Lustgarten Labs program. Lab stands for Lustgarten Advancing Breakthrough Science, and that's exactly what they're doing. Our work is driven by science, and now more than ever, we're looking to the future in ways we couldn't have imagined just a few years ago. And of course, none of this would be possible without our donors. The Labs program is made possible in part thanks to the generous support of the Burns Schwartz Family Foundation, the Wassily Family Foundation, the Warren Alpert Foundation, and we are also grateful for the ongoing support of Ipsen. And now it's truly my pleasure to introduce my colleague, Andy Rakeman, Lustgarten's Vice President of Research. Andy, today's program, months in the making, is exciting for so many reasons. It's our opportunity to showcase the scope and breadth of our ongoing efforts and to highlight the vast talent that makes up our lab's ecosystem.
Yeah, Linda, I couldn't agree more. I'm super excited for the event today. I think it's going to be fantastic. Like all of our Lustgarten Lives, we're going to hear about some great research. We're going to hear about some really exciting results. We're going to meet some brilliant people. But what's new on this one that I hope our listeners take away from it is they're going to get a little bit of a peek behind the scenes and how does the research happen? Who are the real folks driving what's coming forward and what kind of passion and talent and creativity goes into that? So as you mentioned, all the people we're going to hear about are postdocs. Scientists love some jargon, and we also love some good abbreviations. So for people who don't know, a postdoc is short for postdoctoral fellow. These are people who have completed a doctoral degree, so either an MD, a PhD, or for the overachievers, both, and are now looking to launch that independent part of their research career. So they're working in a lab where they get support to take all of the different skills and talents and ideas that they've developed in their doctoral training and apply that to a new problem and create an independent line of research that they can hopefully take forward from there. So as you might imagine, it's a super exciting time in somebody's career, a time of a lot of passion, a lot of creativity, a lot of camaraderie with the folks that they're working with in the lab. And we had a little bit of a preview. I think that's really going to come through in the presentations that people hear today. So the six folks we'll hear from, they have a lot of things in common. They're all really dedicated to pancreas cancer research. They're all brilliant. And they're all, as you mentioned, working in one of our six labs. So people who have been tuning in to Lustgarten Live all along should be familiar with our labs program. This is the crown jewel of the Lustgarten research portfolio. It allows us to, it's a way that we give support to really high performing labs that allow them to take on the most challenging problems in the most fearless way possible. So to really take on the biggest challenges. But more than that, it lets the leaders of these labs create these environments that become sort of a nexus, that attract top talent, that build a community really focused around pancreatic cancer research. So you'll see some of the talent that comes into these sites when you hear from the postdocs that we're going to hear from today. You'll hear about a lot of different topics, but these are all topics that if you've been following with Lustgarten, you hear about before: immunotherapies and vaccines, early stage clinical trials, cancer early detection, pancreas cancer early detection, or that work to fundamentally understand what causes and what drives pancreatic cancer, what's in that biology behind it. So again, I'm going to stop. Otherwise, I'm going to fill up the whole hour telling you how excited I am. But since this is a bit of a different format than what we're used to, maybe, Linda, you can kind of go over what to expect in terms of the process for today.
Great. Thanks, Andy. That was a great overview of how important our labs program is. So this is how this super exciting sprint will work. Each of our postdocs will have 4 minutes each to explain their research in a way that's engaging, understandable, and impactful. And at the conclusion of the presentations, we will invite you, everyone watching, our Lustgarten community, to cast your vote for the project that excites you the most. So listen carefully, stay tuned, you're in for a great treat, and Andy will introduce you to our postdocs.
All right, thanks, Linda. So here's our lineup of stars for today. From the Vogelstein lab at Johns Hopkins, we're going to hear from Jacqueline Douglas. Dr. Douglas earned her MD and her PhD from Johns Hopkins University, where her doctoral research in the Vogelstein lab focused on antibody engineering and cancer therapeutics. She's completed an internal medicine residency at Johns Hopkins and is a medical oncology fellow, as well as a postdoctoral researcher in the Vogelstein lab. From the JAX lab at MIT, we'll hear from Hannah Hong. Dr. Hong received a PhD in immunology from the University of Michigan. She's currently a postdoctoral fellow at the Koch Institute at MIT, where she's looking at immune dysregulation in pancreatic cancer. And from the Tuvison lab at Cold Spring Harbor, Jonathan Kasten and Dr. Kasten earned a PhD in genetics and genomics from Duke University, where he studied immunotherapy for glioblastoma, obviously not afraid of taking on the hard cancers. So then he turned his attention to pancreatic cancer and works in the Tuvison lab on new approaches for early detection. And from the Jaffe lab at Johns Hopkins, Dr. Liang received an MD with distinction in research from Stony Brook University School of Medicine and completed an internal medicine residency and chief residency at Johns Hopkins Bayview, and is currently a medical oncology fellow at Hopkins and does research focused on immunotherapy approaches and early stage clinical trials in pancreatic cancer. And from the Wolpen lab at Dana-Farber, Anders Ullman, and Dr. Ullman earned his PhD in pathobiology from Brown University and is a postdoctoral fellow in the Wolpen-Nowak lab at Dana-Farber. And he uses some really advanced techniques that he'll describe more about that give us spatial information and multiple kinds of omic information, so genomics and proteomics and everything together, to look at the impact that pancreatic tumors have on adipose and muscle wasting, so cachexia, that severe weight loss that goes along with pancreas cancer. And finally, from the Shaw lab at the Salk Institute, Shira Yamtubian, and Dr. Yamtubian received her PhD from Weill Cornell, where she studied mechanisms of metastasis in triple-negative breast cancer. And she's now working at the Shaw lab at the Salk Institute, looking at genetic factors that influence pancreatic cancer initiation and progression. So everybody take a look at these fine faces and these names. These are names that you're going to hear more about in the future in pancreatic cancer research as they continue to have an impact. And we're really excited to hear about the research that you're doing and see what's exciting you most. I know I'm going to need more than one vote at that poll at the end of the day today, but looking forward to it. So without further ado, let's jump into the presentations. And the first person we're going to hear from, we drew straws, so these are in no particular order, is Dr. Douglas. So as we give him a moment to get reset, we will start the countdown on your 4 minutes, Dr. Douglas, once your slide comes up. Excellent.
Thank you to the Lustgarten community for the opportunity to share our work today. Pancreatic cancer is one of the most aggressive and difficult cancers to treat. Even today, fewer than 1 in 10 patients live 5 years after diagnosis. And one of the biggest reasons for this is that cancer cells look a lot like normal cells. So developing therapies that kill only the cancer while sparing healthy tissue remains a huge challenge. But there is one key difference we can use to our advantage, and that's mutations, or changes in the DNA of cancer cells. This is important because inside any cell, DNA gets expressed as proteins, and those proteins are broken down into tiny fragments called peptides. Some of these peptides get displayed on the cell surface by molecules called HLAs, sort of like tiny flagpoles that hold up small protein flags. A normal cell would express only normal peptide HLAs. However, in cancer, mutations in the DNA result in mutant proteins and thus mutant peptides, which can result in mutant peptide HLAs on the cancer cell surface. You can think of this as a red flag, a marker that says something is wrong here. These mutant peptide HLAs are truly cancer specific and represent ideal targets for precision therapies. Sometimes patients do have T cells that recognize these red flags, but for most, especially those with pancreatic cancer, the immune system can't mount a strong enough response, in part because the pancreatic cancer is good at hiding from immune attack. And that's where our project comes in. We're designing small proteins called antibodies that can recognize these mutant peptide HLAs, essentially tiny homing devices that can zero in on the red flags unique to cancer cells. We're particularly focused on mutations in a gene called KRAS, which drives the majority of pancreatic cancers. Because KRAS mutations are so common in this disease, targeting them could be a powerful and specific way to attack pancreatic cancers. Once we have antibodies that recognize the KRAS mutant red flags, we engineer a type of immune cell called a T cell to express a special type of T cell receptor called a TRuC that incorporates our antibodies. On their own, T cells can't always tell which cells are dangerous, but by equipping it with one of our antibody-based homing devices, the T cell can now spot cancer cells waving those mutant KRAS red flags. When a TRuC T cell finds a target, it locks on and delivers a precise immune strike, destroying the cancer cell while leaving healthy ones alone. We're also working on how to improve how we go about finding the best antibody homing devices. Traditionally, antibodies are chosen based on how tightly they bind to a target, but binding alone doesn't always predict function. Some that bind well don't actually activate T cells effectively. So instead, we're developing a functional screen using a library of TRuC cells that lets us test millions of different antibodies all at once. Instead of picking candidates just because they bind well in a dish, we're letting the T cells themselves tell us which receptors actually work, which ones recognize the right flag and trigger an immune attack. This approach could help us identify the most effective TRuC designs and move us closer to therapies that can finally break through the immune resistance seen in pancreatic cancer. So to summarize, pancreatic cancer is very difficult to treat, partly because it hides from the immune system. KRAS mutations are found in nearly all pancreatic cancers and raise unique red flags on the cell surface. We're designing TRuC T cells to specifically kill mutant KRAS-bearing pancreatic cancers, and a TRuC library functional screen may help us identify the best antibodies to guide future therapies. If successful, this work could pave the way for a new generation of cancer immunotherapies, ones that recognize cancer not just by where it grows, but by its unique genetic signature. Thank you.
Thank you, Dr. Douglas. That was fantastic and really inspiring of how many different elements you're able to bring together, and really the way you're thinking about taking the complexity of the immune system and using that as a way to turn that into specificity for how we approach pancreatic cancer. So thank you for that. Very exciting work. And next up, we're going to hear from Hannah Hong from MIT, who's going to continue a little bit on the theme of immune approaches in pancreatic cancer, but talk about that in the context of some other treatments and how we might use these together. So Dr. Hong, welcome to the screen, and we'll start your time once your slide is up.
All right, thank you so much. I'm really excited to share some new data with you all about how we're thinking about new cancer vaccines and how they may be utilized in combination with a targeted therapy that can inactivate the most common genetic mutation in pancreatic cancer called KRAS. Okay, with that as our launching board, the long-standing goal of the JAX Lab is to better understand how pancreatic cancer progresses and evolves. The vast majority of pancreatic tumors are initiated by the activation of the KRAS oncogene, and over time, this leads and allows these cancer cells that harbor this KRAS mutation to grow uncontrolled. This leads to late-stage disease where the majority of patients are diagnosed, and there remains an unmet need to develop better, more effective therapies at this stage. The development of small molecule inhibitors that can target this mutant form of KRAS has shown to be potent and can lead to great tumor regression in both preclinical and clinical trials. Because of these results, there's great optimism that this could lead to a radical change in the way that we treat pancreatic patients. However, preclinical data suggests that these KRAS inhibitors as a monotherapy may not be enough to eliminate or clear tumors completely, and this could lead to potential disease relapse. Because of this, we and others have started to think about combinatorial therapies that can lead to complete tumor clearance as a way to limit tumor relapse. This leads me to my second therapy, cancer vaccines. So recently, cancer vaccines have been shown to effectively clear or control tumor growth following surgical resection in a subset of pancreatic cancer patients. One barrier to the application of these vaccines is that it will likely only be effective when the tumor burden is small, so just following surgical resection. So, as a proof of principle, to see whether we can extend the benefit or use of these cancer vaccines, my project aims to identify whether we can combine a KRAS inhibitor, which we know will lead to low burden disease, along with a cancer vaccine, and ask, is this the right therapeutic window in which we can harness the immune system to target the persisting cancer cells? So how do we test this? The immune system is only as good as its ability to detect and distinguish a malignant cell versus a healthy cell. And pancreatic cancer is particularly good at evading immune detection. And so, as a proof of principle, the way that I've decided to look at this is using a unique mouse model of pancreatic cancer in which mice harbor pancreatic tumors, in which we've engineered these tumors to express a cancer target. By doing this, we can make cancer vaccines very similar to the ones that are used in the clinic to administer and then track the immune response induced by these vaccines and see how well the anti-tumor immune response can then target our cancers of interest. And so in this experiment, what I'm showing is a survival curve. And so in the black graph or bar graph, you can see that the animals with no treatment. So these are our controls. They had pancreatic tumors, and we can see nearly 100% of them succumb to the disease around day 50. When we treat these animals with a KRAS inhibitor, we can extend that survival. But when we layer on a cancer vaccine along with our KRAS inhibitor, we can extend that survival even more. And most excitingly, if we focus in on this tail of the green graph in which we had the combination, we can see that roughly 50% of these animals were still alive by day 200. In fact, they're still alive today, and they're living in our mouse facility with no detectable tumors, suggesting that this combination effectively cleared those pancreatic tumors. And so we're really excited about this data, but we're also thinking about next steps. How can we increase the survival from 50% to perhaps 100%? So what I'll leave with you all today is our next step and what we're thinking about in terms of combining an additional therapy with what we have so far. So we know that cancer vaccines can educate the immune system. We now want to ask, can we amplify that response by using additional immune therapies such as checkpoint? An example could be anti-PD-1. So I'm really excited for this data to mature in the direction of this project. And again, I want to thank the Lustgarten community and foundation for their support. I also want to acknowledge my advisor, Tyler Jacks, for his support of this project. And lastly, I want to mention that this is a huge project. It's a collaborative team effort. And so it is a collaborative project between myself and a fellow postdoc in the lab, Isabella Ebevold, as well as a lot of help from my undergrad, Megan Zhu, and technicians Wang Ying and Kronrad. Thank you.
Thank you, Dr. Hong. That was excellent and very exciting to see. And it's amazing to think about as we add all of these additional tools to our toolbox in the fight against pancreatic cancer, the potential there is in combining them, and seeing that long green tail for your mice is what we all want to see at the end of the day. But moving on from there, so we know that there's work happening, new kinds of treatments coming to the fore, but we also know we have to do better to find pancreatic cancer patients earlier. So next, we're going to hear from Dr. Jonathan Kasten, who's going to tell us about the work that he's doing in the Tuvison lab at Cold Spring Harbor to develop blood tests that'll help us to find pancreatic cancer patients earlier. So Dr. Kasten, please take it away.
So first, I wanted to say I'm very excited to present my project to the Lustgarten community. So my work focuses on identifying blood biomarkers that may aid in the earlier diagnosis of pancreatic cancer. Why is this important? As you can see from this graph, those who are diagnosed when the disease is still localized have a substantially higher 5-year survival rate compared to those diagnosed with later stage disease. As I'm sure many in the audience know, a particularly evil aspect of this disease is that far too many people are still diagnosed at these late stages, leading to poor overall survival rate. So my goal is to hopefully shift this diagnosis spectrum to finding more individuals with early stage disease and therefore significantly improving the survival rate. So before I dive into my approach to tackling this problem, I want to emphasize 2 key aspects of any diagnostic test. First is what is known as the false positive rate. This is the rate at which individuals who do not have pancreatic cancer test positive with the test. The second is the false negative rate. This is the rate at which individuals who do have pancreatic cancer test negative with the test. These 2 metrics are related to one another, but I have been largely obsessed with false positivity during my postdoc. This is because, fortunately, pancreatic cancer is a rare disease, which means even an extremely small false positivity rate will lead to far more positive tests for those who do not have pancreatic cancer as opposed to those who do. So my work is trying to address how to find biomarkers that minimize false positivity as much as possible. So to introduce how I'm approaching this question, let us take a step back. Cells, like us, have to communicate with their neighbors. They do so by releasing certain proteins that will tell other cells what to do, from mounting an immune response to repairing a wound. Cancer cells are no different in this regard, and as depicted here, they can tell other cells called fibroblasts to make their bed or create an environment that allows cancer cells to persist, proliferate, and thrive. As you can imagine, if we can begin to understand the proteins or words communicated by cancer cells, we can begin to try to find ways to interpret messages that only a pancreatic cancer cell may send, much like a codebreaker may do. The problem with using protein alone is that many cells in our body communicate using the same proteins, which often confounds using them as diagnostic markers. Going back to my obsession with false positivity, this is a big issue. To address this issue, I'm approaching the problem by leveraging the fact that cancer cells have altered biology from normal cells. What does this mean? It means that cancer cells have often changed their behavior in many ways that distinguishes them from healthy cells, including how they modify proteins. These modifications include chemicals added to proteins like phosphate, which can alter the function of proteins, splicing, which can physically alter the sequence of the protein, and sometimes unique sugars that are not typically found in healthy individuals. One of these sugars is one that many of you may know called CA19-9, which is commonly used to monitor patient response to therapies. Unfortunately, CA19-9 is produced in other conditions such as pancreatitis, which prohibits its use as a diagnostic tool. However, the idea is that by looking for proteins that contain these modifications that are altered in pancreatic cancer, we will be able to substantially lower the false positivity rate of these proteins and sugars like CA19-9 as biomarkers. So to approach the problem, I've introduced a system into a large panel of patient-derived pancreatic cancer cells and organoids to track which proteins released by pancreatic cancer cells can enter and persist in the bloodstream of mice. Using these same cells in a dish, I've identified which of these proteins are potentially modified with these unique sugars I was talking about. Moreover, with the help of collaborators at Cold Spring Harbor, I have generated antibodies that recognize the specific sequence generated by the splicing of these proteins. All in all, I have identified about 90 exciting modified proteins that could prove to be robust, specific blood biomarkers for pancreatic cancer detection. I'm currently working on further validating these modifications and will shortly begin testing these protein modification combinations in patient samples to generate a discrete toolkit suitable for early detection. So the ultimate goal is to use these modified proteins in the blood of individuals to refine the protein code we are cracking from I am a cell, which is unfortunately not sufficient, to I am a pancreatic cancer cell, providing us an opportunity to diagnose individuals at earlier stages and substantially improve the survival rate for those who have this terrible disease.
Thank you, Dr. Keston. I think the need for early detection is something all of our community really understands very well. I think the complexity and why it's such a challenge is something that's often hard to understand from the patient perspective. I really appreciate you outlining how subtle those changes can be between a cancer cell and a regular cell that's important to detect. And one other request for you in your spare time, if you can figure out that signal that gets people to make their beds, I would love to try that out on my kids, make their beds and clean their room. I'll add on that additionally, but thank you very much. It's super exciting. So I think one of the themes that you've heard so far is that there is something different about cancer cells, and it's understanding that biology and what the difference is, what gives us the tools to come in to whether it's to create targeted therapies against KRAS or immune therapies to recognize the cell or to understand how to detect cancer early. And I think that's a theme that we'll continue to hear about as we hear from Dr. Liang from the Jaffe lab at Johns Hopkins, who's going to elaborate a little bit on the kinds of vaccine approaches that they've been using there and the success and the challenges that they face bringing that forward into the clinic. So Dr. Liang, please take it away once you are ready.
Thank you, everyone, for being here today. My name is Kai Liang. I'm a postdoctoral clinical research fellow at Johns Hopkins. I currently work under the mentorship of Dr. Elizabeth Jaffe and her lab, which is supported by the generosity of the Lustgarten Foundation. Today, I want to talk with you about the exciting work our group has been focused on, training the immune system to fight pancreatic cancer. The immune system is one of the most powerful defense mechanisms our bodies have against diseases. In basic terms, it helps distinguish between healthy cells from unhealthy cells and kill those which may have been affected by bacteria, viruses, or even cells that have become cancer. And for decades, cancer researchers have been trying to harness the immune system to recognize and kill cancer cells. And the benefits of using the immune system as a medicine is that it has the potential to work against all cancers at all stages, both early and advanced. It also has the potential of generating immunologic memory to prevent cancer cells from coming back. However, pancreatic cancer, like many cancers, are sneaky. It camouflages itself and hides itself from the immune system. So how do we teach the immune system which cells are healthy cells and which cells are cancer cells? And one major breakthrough came when Dr. Bert Vogelstein and his lab, a lab also supported by the Lustgarten Foundation, learned that a mutated gene called KRAS acted like an on switch for pancreatic cancer to develop and grow. Think of it like a spark that ignites fire. This KRAS mutation is often one of the very first changes or mutations that turns a healthy cell into a cancer cell. They also found that about 9 out of 10 patients, or 90% of patients with pancreatic cancer, had a mutation in KRAS, making this an ideal target. And so with this knowledge, Dr. Jaffe and our lab designed a vaccine targeted against KRAS. And this vaccine is made from KRAS protein fragments called peptides from the 6 most common KRAS mutations. And these protein fragments act like wanted posters to train the immune system, called T cells, to specifically target KRAS and hunt down cells with mutant KRAS mutations. And so our group has been studying this KRAS vaccine in combination with powerful immunotherapy called immune checkpoint inhibitors, which together we believe can work synergistically to train and boost immune cells to go after pancreatic cancer. Currently, we're actively enrolling in several clinical trials with this approach, and while they're still in phase 1, we've demonstrated that this combination is safe and leads to immune activation. Additionally, since KRAS mutations often occurs even decades before cancer forms, we're using this KRAS vaccine in a prevention role for patients with a genetic predisposition for pancreatic cancer and for patients with precancerous pancreatic cysts. So in summary, this is our big picture. Number 1, using our KRAS vaccine to train the patient's immune system to recognize cells with mutated KRAS. And number 2, combining this vaccine with immune checkpoint inhibitors to boost the immune cells with mutated KRAS. And the benefits of this approach are that it's personalized and selective against KRAS. It's applicable to the vast majority of patients with pancreatic cancer and has the potential of generating durable immune responses, which we hope will translate into improved patient outcomes. Ultimately, it's about creating a new strategy, one that we can finally make pancreatic cancer treatable in ways that we never thought possible. A future where the patient's own immune system becomes their most powerful weapon against this disease. And with your support, we can get there. Thank you.
Thank you, Dr. Liang. Again, another fantastic presentation. And I think another one that, again, tells that story of how early fundamental discoveries, when leveraged in the right way, can be turned into potential products, potential treatments for patients, and moving that forward. But it's really based on starting from that fundamental understanding of what drives a cancer cell and what's different and how do we target it, and moving that forward and having access to these kinds of technologies to develop vaccines and other approaches to bring it forward. So our next presentation that we'll hear from is from Dr. Anders Ullman from Dana-Farber, working in the Wilpin-Nowak lab there, who is looking at understanding not only the how the tumor is developing, but what are the impacts that the tumor has on the rest of the body and the biology of the patient, and how can we use that to better understand and better treat the disease. Very exciting work. Please take it away, Dr. Ullman.
Hi, thank you for that great introduction, and thank you everyone for joining today. Pancreatic cancer is one of the deadliest cancers, partly because it is usually detected late after it has both spread and evolved. And detecting a tumor early is critical but difficult, since early tumors are tiny. And even with modern imaging technology, we can't detect such small tumors inside a patient's pancreas. However, early tumors can cause changes elsewhere in the body even before diagnosis. By analyzing CAT scans, our team at Dana-Farber found that we can detect signs of tissue wasting in fat and muscle up to 18 months before a pancreatic cancer diagnosis. This is because acinar cells in the pancreas produce enzymes that break down carbs, fats, and proteins from the food we eat. And when this system is disrupted by a tumor, it causes the body to break down its own tissues instead. With our collaborators at MIT, we are asking the question whether this tissue wasting can be used as a novel early detection method. This approach would let us find and treat patients sooner when their response to treatment is better. The ideal method to detect these kinds of bodily changes would be via easily accessible samples, such as blood, as Dr. Kasten presented on, and stool, where the enzymes secreted by the pancreas naturally end up. But we need to make sure the changes we see in blood and stool truly reflect what's going on inside the pancreas. To answer that question, we're using state-of-the-art spatial technologies. Our lab specializes in tissue-based analysis, which allows us to see microscopic details of the normal pancreatic cells and helps us understand how cancer cells can change the biology of the pancreas very early in the development of a tumor. The first technology we're employing is multiplexed immunofluorescence, which shows us the expressed proteins that make up cells and give them their identity or their phenotype. We've selected marker proteins for a variety of cell populations inside the pancreas, and we can visualize them. In this example, you can see the enzyme-producing acinar cells in orange and insulin-producing islet cells in blue. Using machine learning software, we can quickly map what cells are present, where they are, and if they look normal or not. The second technology we're using is spatial transcriptomics. This allows us to look at the underlying gene expression of each cell, which drives their behavior. If the immunofluorescence tells us what each cell is doing, this tells us why they're doing it. And using this technology, we can compare cells from different areas of the pancreas. So here you can see we can easily tell the different types of islet cells apart, which look the same under IF, but they have different gene expression. We ran a preliminary study using a mouse model of pancreatic cancer, which allowed us to compare pancreatic tissue from tumor-bearing mice to healthy controls of the exact same age. We also developed a very sensitive way to measure enzyme activity in stool. This way, we can directly compare the tissue to stool samples to see if the stool enzyme changes mirror what's happening inside the pancreas. If they do, our stool probes could lead to a novel, non-invasive way to monitor high-risk patients for very early signs of pancreatic cancer. We discovered there is indeed a drop in pancreatic enzyme levels within the normal acinar cells of mice with tumors. This drop was seen both in the pancreatic tissue itself and also in the stool for the 3 major enzymes we tested. And the degree of change in both of these samples was similar, showing that the stool enzyme changes truly reflect the changes inside the pancreas. Next, we'll test earlier time points in tumor development to see how early we can reliably detect these enzyme changes. Then we will move to human samples to determine if these findings are translatable to patients. And that, of course, is ultimately the most important part of our research. One of the major attributes of Dana-Farber is that we have direct patient interaction. And we have a dedicated pancreatic cancer research division, the Hale Family Center, with over a dozen labs. As a result, we have the ability to draw from a large amount of banked patient samples the ability to design new clinical studies with a large local cohort of patients, and the ability to collaborate with experts in the field under one roof. With the support of the Lustgarten Foundation, we hope to change the status quo of diagnosis and reach patients earlier, while at the same time developing a deeper understanding of how pancreatic cancer develops and causes tissue wasting. Thank you so much for joining.
Thank you, Dr. Ullman. Again, I think this is a topic that every patient or somebody who took care of somebody with pancreatic cancer understands very well, this severe muscle and fat wasting and cachexia that happens in the patients. And it is, definitely inspiring to hear how that kind of observations is being learned from to teach us how to better understand detection and treatment of the disease going forward. So again, exciting work. We're not done yet. We have one more to hear from, and we will bring up Dr. Shira Yamtubian from the Salk Institute, where she works in the Shaw Lab, who's going to tell us the work that she's been doing of using genetics of pancreas cancer to also uncover what are the kinds of mechanisms and the ways that we might go in to alter the course of this disease going forward. So Dr. Yamtubian, please take it away.
Hi, everyone. I'm so thrilled to be here with the amazing Lustgarten community and to represent the impactful research we're doing at the Salk Institute. So the average lifetime risk of developing pancreatic cancer is about 1 in 64, but that risk can increase due to a variety of risk factors, one of which is family history of pancreatic cancer. In fact, around 10% of pancreatic cancer cases are hereditary. That means that for every 10 people diagnosed, 1 likely carries an inherited genetic mutation that increases their risk of developing the disease. It is important for us to study these inherited genetic mutations because they give us insight into what cancer cells need to initiate and progress in the pancreas. We now know that most of us have hundreds of tiny precursor lesions in our pancreas. These are small abnormal clusters of cells that have begun to change but are not yet cancerous. Thankfully, the incidence of one of those precursor lesions becoming a tumor is very, very rare. So one of the biggest questions in the field is, what makes a precursor lesion become a tumor? What are the factors that reprogram these cells to become tumors? If we can figure that out, we could develop better ways to detect, prevent, and treat pancreatic cancer. One of the strongest inherited risk factors is a mutation in a gene called LKB1. When LKB1 is mutated, it loses its function and causes a condition known as Peutz-Jeghers syndrome. Sadly, these patients have more than a 50-fold risk of developing pancreatic cancer. Despite its clinical significance, we don't fully understand how losing LKB1 function drives tumor growth and why people with these mutations are so much more likely to develop pancreatic cancer. So what is LKB1? It's a kinase, which is a type of protein that acts like a molecular switch, turning other proteins on or off. In the case of LKB1, it activates proteins that keep cell growth in check and maintain normal metabolism, acting as a tumor suppressor. But when LKB1 is mutated, that control is lost. In lung cancer, our lab has shown that losing LKB1 activity promotes tumor growth by driving unchecked cell growth, altering metabolism, and based on my research that will be published soon, helping cancer cells evade the immune system. Building on these insights, my work in the Shaw Lab now focuses on how mutant LKB1 drives pancreatic cancer. Using a combination of sophisticated mouse models that mimic pancreatic cancer development from early lesions to full-blown tumors, along with high-resolution tools to track cellular and molecular changes, we discovered that LKB1 does most of its tumor-suppressing functions through a group of proteins known as the Syk family. When LKB1 is mutated, these Syk proteins stay inactive, which reprograms the cancer cells, and very early on, before there's even a visible tumor, begins recruiting tumor-promoting cells like macrophages, neutrophils, and cancer-associated fibroblasts. My research now focuses on how the LKB1 Syk axis recruits these tumor-promoting cells and creates an environment that supports tumor growth. LKB1 mutations are one of the strongest inherited risk factors for pancreatic cancer. Yet until now, we didn't know how they drive the disease. Our work thus far has uncovered the key proteins that carry out LKB1's tumor suppressing role, bringing us closer to understanding what pushes early precursor lesions to becoming cancer. Ultimately, by studying these inherited genetic clues, like LKB1, we hope to crack the pancreatic cancer code and open the door for early detection, prevention, and treatments that could save lives. Thank you.
Thank you, Dr. Yamtubian. And I think you summed up the mission of the Lustgarten Foundation very much at the end there. It's uncracking the code to get us to better detection, better treatments, and lives, not lives being saved. So thank you very much. And I agree. I think this data that you alluded to, that these precursor lesions are much, much more common than we thought. And after a certain age, all of us are walking around with them, but it's very rare that they progress. And, really understanding that switch very early is going to be key for that kind of next stage of what we think we can do in pancreatic cancer. So this was very exciting. I heard a lot of things that I'm thrilled about. It's work that even I've been following through our support of the labs and hearing about at meetings, but it's great to hear about it in this kind of a format and to see you all together. I think, Linda, now is the time where we're going to turn it over to the audience. Is that correct?
Yep. So now it's time for our audience. I hope you enjoyed these presentations. And I know I am so excited to meet these young leaders, and I'm super excited by their projects. And I hope the audience has a better understanding of why this research is so important, why it takes so long, but also because of the iterative process of what these labs do. I hope you understand more of the ecosystem that the Lustgarten Foundation supports. And you know the importance of all the work, but you have to pick one. So before you vote, and we'll put that poll up in a minute, Andy's going to remind everybody of the postdocs' names and their project titles.
Yep. And so first project we heard about was outsmarting pancreatic cancer by turning mutations into targets, and we heard about that from Dr. Jacqueline Douglas. We next heard from Hannah Hong, who told us about Stronger Together, a new duo in the fight against pancreatic cancer, like a good superhero duo coming through. We then came back to Long Island, and Jonathan Kasten, who presented his work on developing a blood test for early detection of pancreatic cancer. Then went back to Baltimore to hear from Dr. Liang about KRAS vaccines, training the immune system to fight pancreatic cancer. And then from Anders Oman, who told us about spatial multiomics characterization of exocrine dysfunction in early pancreatic cancer development. And finally, from Shiri Yamtubian, who presented inherited clues to crack the pancreatic cancer code. All wonderful. I think I'm not voting because I'd have to have 6 choices here. It's all really thrilling and exciting. And I think that the poll should be going up to give people the opportunity to vote, but I'm not going to be able to resist while I have everyone here and such brilliant minds. Beyond the exciting science, I heard a lot of themes that cut across all of your projects and all of the work that you presented, from things being made possible because of access to exciting models and experimental paradigms, the bringing together of findings from other areas or paradigms to bear on pancreatic cancer and how that can advance us. The importance of collaboration to get things done in the lab. So maybe, I don't know if I'm going to put somebody on the spot, but maybe, talk if somebody wants to make a comment on, what is special about the environments that you're working in that allows these disparate things to come together. And, are there ideas that come up that you wouldn't have thought of on your own, but because you're in an environment with all these other researchers around you, that really helps to spark new thoughts and new ideas? I don't know if anybody wants to take it away, or if I can pull out the finger of doom. But maybe, I know, Hannah, you talked about the collaboration that you had within the lab. Is that something that's really, unique about the environment that you're in that allows you to be inspired?
I think, yes, it's a little bit about the environment. We do have a concerted effort in the lab to focus on pancreatic cancer. And so there are a group of postdocs that are just narrow-minded, thinking about what's happening, what can we do about it. Isabella and I are also we're immunologists in training. So I think she's also my baby. So that was also very easy for us to just always talk about immunology in the context of pancreatic cancer. And this was like an organic collaboration that we were both very interested in pursuing. But then also I think at the Koch Institute and specifically in Tyler's lab, this lab has been established for so many years that he's seen it from the last couple decades and knows, and there's a rich knowledge in this. And so I think a lot of that is also born through being very invested in understanding pancreatic cancer. So a little bit of both within the lab and also the collaborations that span that.
Yeah,
And maybe go ahead, Anders.
I would echo that. We have one of the great things about our institution is that we have a lot of collaboration within the Hale Center, and like I was saying earlier, we have the ability to work with experts in a particular topic, and we have the ability to specialize. We also have people that are able to span multiple topics and really understand what's going on. So it's great to have within an institution people that are able to specialize in particular topics and also people that can cross-talk. We have that at Dana-Farber. We also have a lot of collaborators, for instance, at MIT and in the Mass General Brigham system. So if we're not limited. We don't have to be a lab that does everything all at once. We can work with people that are mouse model experts. We can work with people that are data science experts, and we can do what we do best.
Thanks. And maybe for Jonathan and Shira, because both of you talked about work using either very specific mouse models that have been developed or these human organoid models. So maybe if you can have a comment or two to help our audience understand, when a new model becomes available, how much does that really open up in terms of what's possible to understand, and how do you think about your research differently and when based on the availability of these kinds of research models?
Yeah, so I mean, all models used in science are going to be imperfect, and a lot of science is dedicated towards making better and better models. A lot of models will be better suited for answering some questions versus others, and having an entire toolkit of models is really ideal for making discoveries that are robust and hopefully translatable to patients. So having things like organoids, which did not exist 10 years ago, makes a huge difference for us, because we can do a lot of things with organoids, like, for instance, when you implant them into mice, they tend to form tumors that are more similar to what you would see in a patient as compared to the typical cell lines we've used in the past. And so that's just one example of how useful they've been, as well as generating just an array of organoids that are derived from patients allows us to understand the heterogeneity among pancreatic cancer patients that we may not have appreciated before.
Yeah, to piggyback on that, it's really exciting when, someone publishes a new model. One of the things that, two things that we're constantly thinking about is, one, does it help us answer the question that we're looking for? Does that model really recapitulate the human condition and what we're trying to ask? And the second thing is time. Not only just because we're trainees, but, we want to start answering these questions quickly. So personally, I actually went to the Tuvison lab where Dr. Caston's at and learned a pancreatic cancer model that, turned, a model that would take us about 3 years to make, I can now do in 4 months. So it's really great to learn from other people what kind of models they're using and when they publish a new model.
Oh, that's exciting, and it's always great to hear about that kind of collaboration happening across the institute. And I know this has been my experience coming into the pancreatic cancer research community. I'm sure it is for everyone else here. It's a special group of researchers who are incredibly collaborative, incredibly open with their data and their resources, and knowing that this fight is really bigger than any one individual or one lab, and we need to be in it together. But my last point, and I know the voting is winding down, but there's one thing I want to bring up, and I'll put our MDs on the spot here. for Jacqueline and Kai. I think everybody talked about, ultimately the plans for translation and highlighted some access to, human samples and human tissues are getting into the clinic. And I know you two sit on both sides. They're seeing patients as well as working in the lab. And, I think we often hear a lot about the importance of moving from the bench to the bedside and moving things forward to the clinic, which is the ultimate goal. But, do you have an opinion of how important is it for folks like you who, for clinicians to really understand the research before it is available and understand what's coming? And, how is that interaction from the clinicians back to the researchers? Is that something that is happening or that we need to really focus more on?
Yeah, I think it's very important to have people who both see patients in the clinical setting and also are involved in the research, because there's so many like practicalities of taking care of patients that we need to be thinking about when we're developing these therapies. So I think it's absolutely critical.
Yeah, I think, in the clinic, everything that we do is evidence-based, and using the basic science, the translational science to really inform the decisions, to tailor in treatment for patients, understanding what has worked in the past, what hasn't worked in the past, and moving forward. And I think, and from the research perspective, as researchers, our call is that our science isn't complete until it's communicated. So us as physicians being able to translate a lot of these concepts for patients and make them tangible is also part of our mission and responsibility as physicians.
That's fantastic. And again, thank you to everyone. This has been a really exciting afternoon hearing about all your work. And like it is, like I said before, it's really amazing to see all the progress that's happening. But Linda, I don't know if I'm sure you feel the same way, excited about what it is, but I think we're also have some more business to take care of here. Right?
So it looks like that Anders Oman from Dana-Farber is the pretty clear leader of the pack. And if you add up our early detection with Dr. Kasten and Dr. Oman, that's 62% of our viewers. So I think it's clear that what the Lustgarten community is looking for is a way to detect this deadly cancer much earlier. And that's obviously not to say that we are not thrilled and excited by everyone else's work with KRAS, with training the immune system, outsmarting pancreatic cancer by turning mutations. All of your work is terrific. And what I really hope is that what it shows our community is the importance of science, the importance of research, and the importance of maintaining our young researchers in this ecosystem of pancreatic cancer. So I hope our community will share what they've learned today with their friends, with their families, explain to them the importance of supporting science, and of course, the importance of supporting the Lustgarten Foundation. And we're thrilled that all of you were here to learn from these exciting young researchers who are the next leaders in this field. And the Lustgarten Foundation is thrilled to be providing an honorarium to all of our participants here today. And we'll be looking forward to our next Lustgarten Live, which I'm sure our community will be thrilled to learn. It's Unlocking Early Detection. And the title is Unlocking Early Detection from Precancerous Clues to AI-Driven Discovery. And that will be taking place on November 19th. We will be welcoming Dr. Laura Wood, who's an expert in pancreatic cancer pathology. And we'll be welcoming Dr. Elliot Fishman, who is the true pioneer in using artificial intelligence to revolutionize cancer imaging. And together, they'll explore how cutting-edge research is changing what's possible in early detection. So I hope you will check your inboxes. Make sure you sign up for our next Lustgarten Live. Thank you for your participation in our poll. Thank you for listening to our amazing young researchers. And thank you for your support. And we will see you all next month for our next version of Lustgarten Live. Thanks, everybody.